Hello everyone, my name is Océane Cassan. I am a postdoc in the Montpellier Computational Regulatory Genomics team. This talk will cover a new approach to optimize data integration in regression-based gene regulatory network inference in Arabidopsis thaliana. A long-standing objective of systems biology has been to learn the dependencies between genes in the context of development or in response to the environment. One of the first types of data available genome-wide has been gene expression data, and it has now become the input of choice for many statistical methods trying to infer these networks. A category of approach that has shown great performance in challenges such as the DREAM challenge is the category of regression-based techniques, and they rely on the assumption that the expression of regulators hold predictive and descriptive power over the expression of their target genes. And they usually decompose the network inference problem as a series of gene-level regression tasks. Once one regression model per gene has been estimated, feature selection or measure of feature importance allows to identify the strongest interactions and then to infer a final gene regulatory network. However, these approaches suffer from some now widely accepted limitations, such as the high-dimensional setting of the regression models, very strong correlation levels among the expression profiles of genes, and in addition, gene expression alone can only give a partial view of the regulation processes in cells. A way to overcome these difficulties is through data integration and the assumption that complementary omic data can bring more causality to gene regulatory network inference. And so in fact, several existing algorithms incorporate prior complementary omic data by prioritizing the use of regulators supported by the new type of omic data in the model estimation. So these new approaches have shown great potential, however, they open new methodological questions that need to be addressed. For instance, the strength of the contribution of the different types of omic data are very rarely finely explored, although this is a very crucial step of the modeling procedure. And when they are finely explored, tuning data integration usually relies on incomplete or imperfect gold standards that might not be independent from the input data. These difficulties in the fair tuning and assessment of integrative gene regulatory network inference led us to define a new criterion to robustly estimate the optimal strength of data integration based on available data. And for the first time to our knowledge, we propose to optimize data integration at the gene level. In order to provide some fairly general results, we investigate this new approach in two common models for integrative regression-based gene regulatory network inference: linear regression in a method named weighted lasso, and nonlinear regression in a method that we named weighted random forests, that are both inspired from existing algorithms. So as a case study, we propose to study the response to nitrate induction in the roots of the model plant Arabidopsis thaliana, with gene expression coming from an RNA-seq experiment that is already published, in which we identified approximately 1,400 genes that respond to nitrate induction. And among these genes, approximately 200 are regulators. On the other hand, we propose to use the information of transcription factor binding sites that we gather from two databases, the JASPAR and the Plant-System databases. And we propose to build a prior matrix that will contain for each pair of regulator and target gene 0 if the motif of the regulator is not found in the promoter of the target gene, 1 if the motif is found, and 0.5 if the motif of the regulator is actually missing from the two databases, which corresponds to a neutral prior. So here is an overview of the integrative gene regulatory network inference method that we use that are based on regression. In short, they both estimate a regression model per target gene, using as predictive variable the expression of the regulators. And so they both prioritize the use of regulators that are supported by a binding motif during model estimation. In weighted random forests, the usual uniform sampling of variables at the regression tree nodes is now weighted so that regulators that are supported by a binding motif are more likely to get picked. In weighted lasso, a generalized linear model is fit under the lasso constraint, and the strength of the lasso penalty is actually smaller for the regulators that are supported by a binding site. Then the importance of each regulator over each target gene can be measured, and we measure it as the normalized loss of prediction performance that we get when we randomize the expression levels of a given regulator on out-of-bag samples. The final step is to select the strongest interactions on the basis of this importance metric to build final gene regulatory networks. In both methods, the extent to which TF binding site information is used during model estimation relatively to gene expression is tuned by the parameter alpha, which is a crucial parameter of the method. At alpha equal 0, gene expression alone is used. At alpha equal 1, gene expression is used, but the inference of interactions is limited to the interactions that are supported by a binding motif. And in between 0 and 1 lies a whole range of possible values for this parameter alpha, and the big question is how to set an optimal value for alpha. And this is the question that we actually try to answer with our new method. The rationale of our approach is that we want to integrate TF binding sites only if they improve prediction performance when used jointly with expression data. So in order to properly estimate the added value of data integration, we started by simulating a synthetic null hypothesis, where the link between the expression profiles and the TF binding sites of the regulators are actually broken, rendering data integration uninformative. We then measure the effect of data integration by several metrics. First, effective data integration, which measures the effective contribution of regulators supported by a binding motif to the prediction of the target gene expression in the regression models. And we measure it as the average rank of regulators supported by a motif when regulators are ranked by their importance value. And so as expected, we can see that increasing the value of the parameter alpha increases effective data integration, both in the true dataset and in our shuffled baseline. We also measure the ability of the regression models to predict the target gene expression on test samples that are actually out-of-bag samples that were not used during model training. And so this prediction error, as we can see for example here in this gene example, is impacted by the parameter alpha, and the trends in the true dataset and the shuffled baseline are actually not the same. Finally, we put together the variation of effective data integration and prediction error in order to find the optimal value of the parameter alpha. And we define it as the value of alpha for which the prediction error is most reduced as compared to our synthetic null hypothesis. In this gene example, we can see that this is kind of the ideal case, where using the information of binding sites guided model estimation toward the selection of robust regulators that provide a better prediction of gene expression on test sample. And this is actually a very nice example of how data integration can alleviate the curse of high dimension and the problem of high correlation between regulators. In other cases, we can see that the effect of data integration is less clear. For example, in the left, the prediction error is most reduced for an intermediate value of alpha that we set to 0.5 with our approach. And on the example on the right, we can see that integrating TF binding site information does not actually impact prediction error that remains stable. However, our approach can still allow data integration given that true prediction error remains sufficiently lower to the synthetic shuffle datasets. And finally, there are cases where data integration is not beneficial to the prediction of gene expression. For example, in these cases, prediction error is the same or is even higher than our shuffled baseline So in this case, our method, we set the parameter alpha to 0. Taken all together, these results and these gene examples show that the effect of data integration are not the same depending on the genes, supporting the use of a gene-level optimization procedure. We then investigated the overall quality of the inferred networks, either in globally optimized networks with the same value of alpha for all genes that are shown in green here, or with our gene-specific optimization procedure that is shown in blue. And we focus on two metrics. First, the median prediction error on all the nitrate-responsive genes, that is shown in the first row for the two models, linear and nonlinear. And what we can see is that using TF binding sites tends to increase prediction error of gene expression on test samples for globally optimized networks, and that our gene-specific procedure offers to minimize prediction error, and this holds for the two models. We then looked at the agreement of the inferred gene regulatory networks with experimental in vitro binding interactions given by the DAPSIC techniques. So we can see that in globally optimized networks, using more TF binding sites, information increases both precision and recall, and that our gene-specific procedure allows to achieve a good precision and recall, although it is not as high as some globally optimized networks. However, we argue that this is a desirable trade-off, where our method can minimize prediction error of gene expression on test sample, that is a condition-specific metric that can be used and applied to all genes, while still providing a satisfying precision and recall on an experimental gold standard. We then assessed the ability of the inferred gene regulatory networks to retrieve important actors of nitrate signaling in the roots of Arabidopsis, which is quite a well-documented response. And so first observation is that regardless of the model or the data integration strategy, if we look at the top regulators in terms of out degree, we can see that the 25 most connected regulators are actually enriched in regulators that are known regulators of the nitrate response. These are shown in orange and yellow here. And more importantly, our gene-specific optimization method uniquely retrieves some major regulators of the nitrate response. That's the case for weighted lasso and the regulator NLP7, which is the master regulator of nitrate early response. And it also puts forward new candidates that remain to be validated. We then looked at the distribution of the optimal value of alpha for all the nitrate responsive genes, and we noticed that for a majority of genes, alpha was actually set to 0, and this is true for both models. So this opens a lot of new questions. For example, we could hypothesize that these genes for which alpha equals 0 might be regulated by condition-specific regulators that need cooperative binding with another regulator that would have a stable expression level. Or maybe the messenger RNA levels of these genes are regulated by post-transcriptional mechanisms, impacting their stability. And these new questions might be answered by integrated new types of omic data in future works. So the main message of this talk is that in order to tune integrative regression-based gene regulatory network inference in a gene-specific manner and in a data-driven manner, we relied on two ingredients. First, using prediction performance of gene expression on test samples. That is a metric available for all genes and that is condition-specific or tissue-specific, and that we propose to prioritize over the use of related or incomplete gold standards. And second, we simulate a synthetic null hypothesis that breaks the assumptions of data integration, and in this setting, we can choose to integrate TF binding sites when their use improves prediction performance over this shuffled baseline. As a result, we show that we can achieve a desirable trade-off between minimizing prediction error of gene expression and a good agreement on experimental gold standards. We also showed that we can retrieve major actors of nitrate signaling in the roots of Arabidopsis. These results hold for linear and nonlinear regression, showing that this approach has some general applicability to different regression contexts. This work could, of course, be improved in several ways. For instance, computation time could be optimized, and the fact that a fraction of binding sites are still unknown from current databases is also a limitation. Hopefully, these databases will grow in the future as they are maintained by the community. This work also opens new questions and broader questions for future research. For example, exploring the differences between weighted lasso and weighted random forests would be a nice way to test the impact of linear and parametric assumptions in gene regulatory network modeling. And of course, it would be very exciting to extend this work to new organisms, and that might require the addition of new types of omic data to scan and answer regions for TF binding sites as well. I will conclude by thanking the conference and NetBio organizing committees for their work and for inviting me as a speaker. And I'm also very grateful to my entire team from the Montpellier Computational Regulatory Genomics team, Antoine Martin from the Institute for Plant Science in Montpellier, and the University of Montpellier and the CNRS for funding my project.